So, hello everyone, we are going to start a new module, that is microbial growth kinetics. So, we will first quickly summarize the last module, that was applied enzyme catalysis. So, where we discussed types of enzyme immobilizations, physical and chemical. Then while immobilizing, we see there is a presence of external and internal mass transfer. How to analyze those external and internal mass transfer resistance and calculate the effectiveness factors. Then further we discussed how to minimize those mass transfer effects, and we broadly observed the applications of enzymes. In today's lecture, we are going to discuss the microbial growth. It's a growth curve. What are different factors which affects the specific growth rate or microbial growth? What is generation as well as doubling time, and different methods for measuring microbial growth. So, microbial growth, as the word suggests, we are discussing increase in the number of microbial cell. So, here we are not referring to the size of individual cells, rather we are referring to the number of cells, and typically cells reproduce at an exponential rate under favorable conditions, and as this is increasing with time, so one analyze the cell kinetics, and this is a result of various complicated networks of biochemical reactions and transport process. Typical process is binary fission. So, you can see here 1 is divided into 2. For example, E. coli divides once in every 20 minutes. So, as I told, this microbial growth is dependent on environmental factors. Some of them are nutrients, oxygen, pH, temperature and light. So, what is the carbon source, whether process is aerobic or anaerobic, what is the pH, whether cell can survive at that particular pH or not, what is the temperature, whether it is lower than optimal temperature, higher than optimal temperature, and whether it requires the light intensity, whether this is a phototrophs or heterotrophs. Now, typically growth curve is a representation that depicts the number with time, and typically it follows a sigmoidal curve which have 4 different phases: phase 1, phase 2, phase 3, phase 4. 1, 2, 3, 4. First one is the lag phase, where bacteria or microbe get adjusted to new environment. Why we say new environment? Because there is change from inoculum to production media. It may be altogether different carbon source. And there is change in the volume. Inoculum is a small volume and production is taking place in the larger volume. Then metabolically active but no cell division occurs, because here you just see there is a almost no increase in the cell number, and synthesis of enzyme for physiological activity. So, basically in the lag phase, cells are acquainted to the environmental conditions, and once they are acquainted, then this bacterial population increases exponentially. So, here once it is increasing, we consider the number of cell produced is exceeding the number of dead cells. This phase lasts for longer time based on the type of organism and culture conditions. So, at the end of log phase, you can see the straight line, it it is reaching to the stationary phase. So, here there is no net increase in the bacterial population. So, we say number of cell produced equals to the number of dead cells. An indication of nutrient depletion and accumulation of toxic byproducts. So, there can be different reason to reach to the stationary phase. One of them is nutrient depletion or accumulation of toxic byproduct, followed by next is decline phase. So, it says now the number of cell dying is exceeding the number of cell produced. It means you will see a decline here, and once you are observing the decline, it is called the death phase. So, number of cells decreases. So, you can see once again the lag phase, there is no change in the number. Exponential phase, we say the generation is higher than the death rate. Here we say both death rate and generation rate is same. Here the death rate is exceeding the generation rate. Now, typically when we see the growth, it follows the first order kinetics. So, it says dx upon dt, change in the microbial cell per unit time is directly proportional to the x. What is the initial number of biomass or cells was there? And as I told, this is typically following the first order kinetics. So, we say dx upon dt, and proportionality constant is the mu into x, and this mu is called specific growth rate. We are saying specific because if you rewrite here mu equals 1 upon x dx upon dt. It means growth of biomass per unit mass of biomass, that is the specific. Specific word means per unit mass. So, in this way the specific growth rate is defined. Now, we can go back again here, dx upon dt is mu x. You can quickly solve here to get the value of mu. So, we can rearrange this expression. So, this will be dx upon x is equal to mu into dt. We can integrate this, and once we are going to integrate this, it was dx upon x to dt. If we integrate, we will end up with ln x mu into t. Now, you apply this boundary condition when t equals 0, x equals x naught. So, integration constant c, if I put the boundary condition, this c equals ln x naught. So, now this will become ln x minus ln x naught mu into t, and this becomes ln x upon x naught is mu into t, and now simply it will become x equals x naught e to the power mu into t. So, in this way, this we calculate at any given time, if number of biomass x is known, we should be able to calculate the mu, because here there is only one unknown, that is mu. So, only one equation will be sufficient to find that value. So, once we know this, we can quickly calculate the doubling time. So, it says x upon x naught, or you can write either way, it says ln x upon x naught equals mu into t. Doubling time means x becomes twice of x naught. So, it will be simply ln 2 is mu into t will be replaced by td, doubling time. So, it says td equals ln 2 upon mu. So, in this way you should be able to calculate the doubling time of a particular microbial strain. Now, what are the different factors which affect the specific growth rate? Nutrients, it is very important because it gives the energy for the growth. So, depending on the nutrient, limiting nutrients, it slow down the growth, and finally, whatever substrate will give, that biomass will utilize for its growth, and also it may utilize the same for the product formation. So, basically it determines the yield. Yield we will be discussing. So, it says yield is y x upon s. So, how much biomass is produced per gram of substrate utilized. Temperature, as I told, all the biochemical reactions are catalyzed by the enzymes, and enzymes perform better at an optimum temperature. So, if there is a low temperature, enzymatic activity goes down. To high temperature, it deactivates or denatures the enzyme. So, we require an optimal temperature. pH, it controls the enzymatic activity and membrane transport, because enzymes are made of proteins. So, they are sensitive to the pH conditions. Inhibitor, we have already discussed different type of inhibition, irreversible and the reversible inhibitions. So, once they are inhibitors, they inhibit the cell growth, and once there is a inhibition of the cell growth, it reduces the value of specific growth rate. Oxygen availability, it depends whether your microbe is aerobic, anaerobic or facultative, and depending on that, if it is aerobic, it requires oxygen. If it is anaerobic, oxygen may works as an inhibitor. Light intensity, as I told, some strains may be phototrophs. So, they require the light for their as their energy source. For example, this algae. So, next we will quickly discuss the generation time. We have already discussed the doubling time. So, generation time, it is time required for microbial population to double in their number, and we are assuming cell division resulting in 2 daughter cells, and if this is there, we also call generation time as doubling time. And as I told, this generation time is dependent on the various environmental factors, as well as it depends on the species. For example, E. coli have doubling time of 20 minutes, while Mycobacterium has doubling time of 12 to 16 hours. Further, for the E. coli in aerobic condition, its doubling time is 20 minutes, but if say anaerobic condition, it becomes close to 2 hours. So, both generation time is dependent on environmental factor and species. So, once we know this microbial growth under ideal condition, it is following exponential growth. So, we say n is n naught 2 to the power n, because we say one cell is dividing into 2 cells, and n is here is number of generation, n is number of generations. So, we can rearrange it. So, this will be n upon n naught equals 2 to the power n. You can write ln n upon n naught equals n ln 2. From here we can get the value of n ln n upon n naught divided by ln 2. Now, we will see one case here, that with time n value is increasing, it is going up to the n equal to 4 generation. Now, depending on the initial number of cell, let us assume initially you are starting with 1. When this n is 1, what will be the number of cells? 2 to the power n, let us calculate. So, this will be 2 to the power 1, 2 to the power 2, 2 to the power 3, 2 to the power 4, and what will be total population? N naught, we are taking 1 into 2 to the power n was 1. So, it will be 1. So, here the same, because we are taking n naught as 1. Once you are taking n naught as 1, so this value will be same, but n naught will have some different value. So, in this way we see, depending on number of generation, what is will be total population. So, for example, if the rate at E. coli divides is 0.5 hour inverse. What is 0.5 hour inverse? Is specific growth rate. So, mu is given here as 0.5 hour inverse. Then estimate the doubling time. We already know doubling time Td is ln2 divided by mu. All the value are given here. You put here, it will be 1.386 hour. One more example we can see. E. coli is introduced in a safe flush condition containing nutrient rich medium. The initial number of viable cell was 10, n naught is 10. After few hours, the number of viable cell is 10 to the power 8. Assuming the binary fission, this is important, calculate the number of generations. We already know the formula n is ln n upon n naught divided by ln2. You put the value, you will get 23.25. So, it is close to 24 generations. So, what is the significance of quantifying the microbial growth? The very first thing, you require the desired number of microbial population for the producing the desired metabolites. So, it directly affects the production yield. Your product may be growth associated or non-growth associated, but in either case you require the cell density, desired cell density. Process control. So, this indicates if there is a good growth, it means your bioprocess is working fine. If there is a deviation from the growth, it indicates there may be contamination, there may be nutrient depletion, or there may be toxins. So, it quickly helps. If there is a no growth, it means some troubleshooting needs to be done. Yeah, this is important for the bioreactor designing, simulation and scale up. We require the value of a specific growth rate, substrate consumption and product formation rate. So, we need to calculate this kinetic parameter, which is required for the bioreactor design and further scale up. In addition, this batch to batch consistency, if you want to ensure the consistency, we need to ensure the number of cells are consistent. This also ensures reproducibility and product quality. Cost efficiency, if you do not have the desired population, your bioprocess yields are compromised, your raw material may be underutilized, it means there are residual substrate is too much. Further, next one is downstream processing. It helps to decide which type of downstream processing you are having. Your product may be intercellular, your product may be extracellular, and which microbes you are using, is it easily separable by filtration or centrifugation. So, in this way we know this microbial growth, it helps in optimization of product yield, process control, kinetic modeling, batch to batch consistency, cost efficiency and as well as downstream processing. Now, next we will quickly discuss how we can measure the microbial growth. So, this is classified as direct methods and indirect. So, first we are going to discuss the direct methods. It measures the cell number or biomass directly. Most popular one is the microscopy count. This is also known as the cell count method. We use this hemocytometer, and the central squares are considered for the counting in a hemocytometer. And once as you are using the microscope, it also gives the information about the size and morphology of the cells. In addition, once you are using the microbes, we can use the stains to visualize the cells. In fact, these fluorescence stains can be used to distinguish live and dead cells. Now, in hemocytometers, typically this size of one square is 1 mm into 1 mm into 0.1. This becomes 0.1 mm cube, and if you convert into the millimeter, so this will be 10 to the power minus 4 centimeter cube or 10 to the power minus 4 ml. So, now you have counted total cell counts in the different squares. You have to also consider the dilution factor into 10 to the power cells per ml, because the volume of one square is 10 to the power minus 4 ml. So, let us say if you have counted 10 squares, you have to divide number of square 10. If you have counted 5, then you have to divide by 5. Next is the visible plate count. What you have to do? You have to dilute, we say serial dilution. Then you have to plate on a solid media containing the desired nutrients for a particular microbe. Then you have to incubate it. Once you are incubating, you will see this visible colony in terms of CFU per ml. And how we calculate the concentration? Number of colonies divided by what was the dilution factor and what was the volume of sample used for the plating. The only challenge here as you have to incubate is this is a time consuming techniques. So, for example, let us say you have plated 1.1 ml 100 microliter and it resulted into 50 different colonies. What was the dilution factor? Was 10 to the power 2. Then I can calculate the concentration, number of colonies 50, dilution factor 10 to the power minus 2. What was the volume of sample I have used? 0.1 ml. So, this I will end up with 50,000 CFU colony forming unit per ml. And next method is Coulter counter electronic methods. So, it says you are going to have this orifice tube and microbial suspension will pass through it, and orifice tube is designed in such a way that only one cell passes at a time, and once the cell passes, there will drop in the conductivity due to increase the resistance, and based on this measurement one should be able to calculate what was the number of cells. This method is suitable for large microorganisms because some bacteria may quickly pass through this orifice tubes. Now, next we will quickly discuss the indirect methods. So, here this method measures parameter that can be correlated with growth, and here the parameter are turbidity, biomass and metabolic activity. turbidity is very easy, you have to measure this optical density. You have to use a spectrophotometer to measure the optical density, and typically we measure the optical density at 600 nanometer in the visual range. And once you know this optical density or absorbance, it follows the Beer-Lambert law. It says A is epsilon C into L. Epsilon is molar absorptivity, C is the concentration which is directly proportional to the absorbance, and L is the path length, and usually its value is 1 centimeter. So, if there is a, as I told, absorbance is directly proportional to concentration. If there are more cells, you are going to have higher turbidity, more light scattering and higher absorbance. So, we measure the optical density, which is defined as log I naught upon I. The typical value of OD, we say OD1 at 600 nanometer. It means there are close to 10 to the power 9 cells are E. coli. It may vary from different strain, but typically we have a correlation between optical density and the number of cells. Next is the dry weight method. In that case, what we have to do, one has to harvest the cell, then you have to centrifuge it to separate the cell, and after centrifugation one need to wash these pellets, further need to be dried, and you have to weigh. And once you are weighing, you know this what was the dry mass and what was the volume of the culture. So, you can say the cell concentration gram per liter or gram per ml. Typically, these cells contain 70 to 90% of the water. So, this dried solid gives the actual solid biomass. In addition to these two methods, turbidity and dry weight, there are other methods like ATP assay. It measures the intercellular ATP using bioluminescence. Other metabolic activity based assay, like one can evaluate the CO2 generation and oxygen consumption. Other method is most probable number test. This is suitable for culture with very less turbidity. So, in the most of the time in bioprocess, we use turbidity measurement because this is a direct measurement. You can measure OD in real time. But when you want to have this dry cell measurement, we have to take out the sample, you have to measure the optical density later on. Other option is you have to dry it and measure the dry biomass. And in the direct method, this hemocytometer is a common one, but if needed we go for this viable plate count. So, with this I will stop this lecture here. Thank you so much.